picture, Pakistani pictures.
Oh, I have so many. I'll start with Gujrat.
Best.
Yeah. So I don't know if that's a good inspiration for Pakistanis to do good work, because that's already there. So maybe we need to motivate you to go out to other places. Right. So today we'll talk about biosensors. It's new module, biosensors. Some basics, some fundamental questions, some fundamental molecules, revisiting some basic high school biology. Some discussion that is already we have done is about DNA microarrays. So what we do, we are looking for the presence of specific nucleic acids. Now, nucleic acids, I'm generalizing, it can be DNA, it can be, like in this case, mRNA, which means now we are looking at messenger RNA. Messenger RNA means the DNA has resulted into a messenger RNA, so that expression of that specific genetic code is taking place, which means, yeah, having the gene is one thing and having the gene expressed is another thing, right? So a person may carry some specific genes, maybe for related to a disease, but those genes never get expressed. So the person never gets that disease. So so we know genetic code is the storage, but the expression is what is more important, which will tell us if that disease has started to show itself or not, or the cells have started to get into abnormal conditions because of because of the expression, right? So expression of that gene is what would the first step would be mRNA, right? So in this case, this example is showing the mRNA. Now, RNA is less stable than DNA. RNA, there's much higher digestive kind of degrading activities against RNA than DNA. So what we do in this case is we get the complementary sequence out of that RNA, and that's what we are looking in this microarray. And here we can compare one expression than the other expression. And we talk about DNA, 4 basic bases, but then the binding of 2 DNA molecule, there are many factors that go into that. We can order DNA. There are synthetic ways where we can make the DNA molecules and we can buy them. But then there are many things that we can engineer into a DNA sequence. So now we are talking about using that as a sensor. We can create secondary structures, like in this case, it's showing a hairpin loop DNA molecule, which means that we are designing a sequence in which we are having some part at one edge, which is perfect complementary to the other edge of that long molecule, which would mean that those 2 edges will come together and find each other, they'll form something like hairpin loop. We play with these tricks where we can then use these molecules to either look for a secondary structure, or we use them to look for the presence of a molecule. And now I'm using the word molecule, not just other nucleic acid, because then there are other subclasses, new classes of DNA molecules. When I say class, the class, it is still DNA, but how we use them. There's a class called aptamer. So aptamer are molecules which show, by experiments, we have found out that they show binding activity with certain proteins or certain receptors or certain organelles. There's no direct way for us to know why it happens, but it happens. There's this example, hairpin loop. If you put kind of a quantum dot on one end and you put a molecule that can quench the light, we call that shape to be a molecular beacon, which is, so we use these, we play with the charge, with functionalization, with adding new groups to increase the sensitivity or selectivity of our biosensor. So DNA has been a major building block of many biosensors that we are using at nanoscale. Now, there are some special properties with DNA, why this has become very prevalent in many biosensing applications. For example, you have seen nanowire example. One of the key paper was about nanowires. So they put DNA molecule on those nanowires to detect another sequence. So the properties that are relevant is by temperature, we can control the binding of two DNA molecules. So we can, if you ramp up the temperature above melting temperature is a temperature, of course, like materials, they melt, which means they change phase from solid to liquid. In case of DNA, we call melting temperature to be a temperature where the double strand molecules start breaking apart into single strand molecules. So melting temperature is the temperature where the probability of double strand and single strand is 50-50. That's what I mean when I say 50% DS, 50% SS, single strand and double strand. So we use this melting temperature property to design our biosensor, where we would know the DNA, one single strand DNA would break apart from other single strand DNA. It can be DNA, RNA. It can be RNA, RNA. There are the new types of nucleic acids. It's called peptide nucleic acid, PNA. There's another type, locked nucleic acid. So biochemists have played and come up with many synthetic nucleic acids as well. But they have the similar selectivity like the living DNA that you would get from a living system. So melting temperature is something we play with. We play with the salt concentration. We play with GC content. So here, if you look at the 5th bullet point, it talks about GC content. So GC is the higher number of GNC, guanine and cytosine, means higher melting temperature. So GC content has been shown to have some interesting roles in the physical properties. So GC, the placement of specific G's and C's, now we know also results into formation of many secondary structures. So which means we can design G and C in a way to make DNA form some structure of our choice. So we engineer these things into our sensors to play or to get the end goal, whatever we want. So above the DNA is the class of molecules called proteins. So proteins are, so DNA, mRNA, tRNA, the nucleic acids gets translated into amino acids. So amino acids are they're very important biomolecules, and all the enzymes in the body are made of proteins. And they're also important structurally because they do the role of receptor molecules. They make up the antibody molecules. So in a more very fundamental level, amino acid has a carboxyl group and an amino group in its structure, and they're then linked with what we call as peptide linkage, which is amide bond, which is CONH, a mixture of COOH and NH group. So we call that a peptide linkage. So if you see the word peptide ever, you would know that we call this bond as the peptide link, right? And then in literature, we have nomenclature that if we are using less than 10 amino acids, we call it a peptide. It's a protein, but depending on the size, we call it peptide. If it's 10 to 100, we call it polypeptide, but greater than 100 is what falls into a protein, right? So protein would have different roles in living systems. A little bit more on amino acids. I wouldn't go in deep into types and their functions, but some things are common. Every amino acid has a carboxyl and amino group. And so if you look at this figure, the bottom part is same in all of them. So the bottom part is same in all of them. It's just the R, what we call side chain. So this red circle is the same. The R is what is different, right? So from an RNA, the machinery that creates polypeptide, the peptide chains, which result into a protein formation, would have the peptide and put the R depending on 3 bases that come in the order. So every 3 bases define, we call every 3 bases as one codon, and one codon is something which would result into those codons. The order of those codons would tell us that what protein molecule needs to be built up, what amino acids need to be built up to create the long chain, which ultimately we call as protein. So now if you look at the simple calculation, 3 bases, so 3 bases make one codon, and there are 4 types of bases, which means there are 64 combinations that these can come. But we only use 20 combinations, right? So we only have 20 amino acids, which means many of those codons do not directly translate into what side chain needs, what amino acid needs to be built in. But they do other functions. Maybe there's some of them are stop codons, some of them are indicative of different things. We don't know many of them, by the way, still. All right, so this is the basic formation of a protein, right? Now, why proteins are important? Because they do everything, right? So they provide structural support, they do protection against outside things, they help transport material from one place to the other, they result into the chemical reactions, they fight against intruders, they regulate the expression of other proteins, or they try to control the expression of things that happen inside. So it's important for us to know about proteins. So it's important for our biosensor, especially nanobiosensors, to know what we are looking for. If any of these functions get disrupted, the cell is diseased. Cell is going crazy. That's where a biosensor is needed, right? Now, functions, reactions, transport, they all are easy to see. But then there are some things which are more profound, like the folding of a protein. The folding of a protein directly results into its function. And there are multiple levels, 4 levels of protein structures. One is just, okay, what comes after what? That's the single length, right? Line-wise, what comes after what? But then there are secondary structures where they may have a helix kind of thing to it. They may be having this plated kind of thing, like you take a piece of paper and you put plates and then you stretch. So they have this kind of structure. These are called secondary structure. So every structure has some function to do in the life of that cell. And then we have tertiary structures, which mean that they are folding onto each other, right? So more of an open fold kind of thing, but the quaternary structure is where they fold on itself. That's the most complex formation. And interestingly, hemoglobin is example of that. Hemoglobin, you know hemoglobin, everybody knows hemoglobin. So what is special about hemoglobin? It has a quaternary structure. We know the structure. Now, if that's, okay, what's the job of hemoglobin? Anybody tell, what's the job of hemoglobin?
To transport the iron body.
Hemoglobin, iron?
Yes.
Right. If it's this, so this is a structure that helps us to do that. If that structure breaks down, what would it do? I think it has to do with oxygen. But it's okay.
Yes.
What if the structure breaks down? But what if the folding is not right? It will not be able to do its job. It will not be able to carry the molecule that it's supposed to carry. So the secondary structure is important. So these shapes and structures define the function of a protein. That's what we use in biosensors as a sensing element. And that binding helps us know whether any of these activities are disrupted, right? Any of these things, any of these functions, if they are disrupted, they would have to do something with either the structure is not folding, or they are not having the right parts that need to go into create that that molecule, that structure of that protein. And we also use that. Then the word here is ligand receptor. So there is a receptor, a protein which is supposed to bind to something, and a ligand is that comes and bind to it. We use their binding known ligand receptors now to functionalize our chip surfaces. We put one thing down and we use that to look for the ligand that's, or we put ligand down and we look for receptor, depending on whatever the application we have, right? So structure and function, the shape gives a specific function to a protein. The presence or disturbance or absence or any of these properties would result into the cell function breaking down, right? So to summarize this, the basic introduction of few molecules, we many times have to see maybe just the expression in cells, because that can tell us the state of the disease or the maybe start of a disease, or maybe the effect of a therapy on a disease, right? So we can many times look from there and even go back and isolate one or few genes that may be culprit into that the feature or whatever the disease happening. We may be able to see if there is a lack of binding, which would mean when a ligand comes and binds with another molecule, we call that signaling, right? So that binding results into further activities. If one thing doesn't bind, the signal pathway breaks down, which means the cell is not doing the function what's good. Maybe it's that what we are looking for. It may be the processes of expression that we are looking for. So all these things have resulted into many different ways. Like we have looked at some examples. This example is where it's from 96, two papers. They came together in one issue of Nature where they used DNA molecules to bring together gold nanoclusters. So they brought gold particles and they coated, so each particle was coated like this. So you see this golden thing, and then there is written SH. SH is sulfur-hydryl group. We call it the thiol group. So thiol group binds to gold. So each DNA molecule had a thiol end group. So thiol end group, once it binds to the gold nanoparticle, and they were floating and they hybridized, what we get is a well-ordered structure like it is shown in this TEM. I think it's a TEM image, 33 nanometer scale bar. So it's one of the earlier examples where they used DNA molecule selectivity to order synthetic structures, to order gold nanoparticles. There is more work, early work, where they used DNA molecules for biosensing, where they were, or molecular electronics even, where they wanted to see if a DNA molecule can be used as an electronic functional part in a circuit, conductivity through a DNA molecule. So many places, either this was synthesized on the chip itself, where they would start with one base and then they would add another base and the chain would form, or they would form it off in a tube or buy it from a vendor and then employ it to put on the surface. So in all cases, the DNA molecule has to have a linker group at the end, right? Like for gold surface, it has to be sulfur group. Again, it's showing the same thing here. And that DNA, modified DNA, requires then careful calculation of what is the surface charge, or what kind of surface, self-assembled monolayer we are putting on the surface. How much is the target concentration we want to look for? So we have to quantify what should be the probe concentration that is enough to detect that target molecules in a given solution. Like in one of the KPI, there was discussion of femtomolar or picomolar target detection, which is very, very small amount. If you remember, 1 molar is the Avogadro's number, right? Anybody remembers what's the Avogadro number?
So 6.02 × 10⁻23.
10 raised to the power 23, exactly. That would be 1 molar, right? And when you say, for example, nanomolar, that would mean 10 raised to the power 23. We have to bring it down to 9 orders less than that. That would make it 10 raised to the power 15, right? 23 minus 9, how much is that? 14. So 10 raised to the power 15, or 14 would be the number of molecules we'd be looking for if you say picomolar, right? So we have to bring that concept. So that density of the surface or density of the molecules would have to depend on what is the concentration we are looking for, how quickly we want that reaction to happen, what kind of buffer. Buffer is, we call buffer as the solution. So which solution we use the molecules to suspend, call it as buffer. So these are now very well-defined SOPs on that, recipes on how we can do that. Many different kinds of DNA molecules have been used, and they've been synthesized and functionalized and quantified in many different ways. All right, so that means we want to use these agents to do sensing, and then sensing has a very huge, especially biosensing. So Bio-Nano has become a foundation of sensing in many, many different areas, whether it's food safety, where we want to look for pathogens in food supply, in vegetables, E. coli, for example. We want to look for bad things in air, in water supply, in post office, in places where people gather, right? Bazaar or places. So we want to make sure there's a way for us to sense and check if there's no bad thing. There is application, of course, in biomedical and medical research, where we want to know the sources of diseases, or we want to know how can we disrupt the pathway of a disease or disrupt the onset of a disease, right? That's where the Bio-Nano is having a big impact. We want to have ways that the patient doesn't have to go to hospital or to a lab, where we say clinical point of care. The doctor's office or a simple clinic should be able to do many more tests at reduced cost, instead of they say, oh, reasonable law. So you don't have to take every patient to Lahore. You know what I'm talking about, right? So in Gujarat, they say it's death sentence, where they say reasonable law, which means, but that testing should be able to be done right there at the clinic. Environment, again, contamination from man-made sources, right? Factories, labs. There's what do you call byproducts of industrial processes. How do we treat them? How do we make sure they don't carry things? Even the what do you call the petroleum refineries? They lots of things that they burn and send out in the air. Public health and safety, we know much more about that now after COVID, that we need tests which can be done on the spot and which can be done without too much training, without having the need of clean environment, right? Without the need of waiting for results for 4 days. So these are all the reasons why we need Bio-Nano, right? So now, knowing or looking at a bunch of molecules that may be interesting and important, looking at the need of why we need to, looking at the reasons why we need to sense them. Now let's look at what are the basic components of a biosensor. A biosensor has to do 3 things. Here there are 2 things which are shown, right? So third thing is just that it has to have a way of telling us the results. But 2 important things is recognition and transduction. Recognition is that it should be able to recognize the right thing that we are looking for. If a test is made for looking at cholesterol, it should tell us only about cholesterol. It should not get, the result should not get fouled or should not get bad because of interaction of another molecule with the component that's recognizing the presence. So we call that molecule or that function of that chip as a recognition element. I see some chat messages. No, not bacteria. I'm talking about toxic things that can come out in the air. I'm talking about the need for sensing those things that may have a biological impact. Right? So now recognition element, you would use this word when you talk about what is relevant medically and how do we achieve selectivity in detecting that specific activity. We call that recognition element, RE. You would see this word RE. RE would mean something that recognizes that molecule or that receptor or that DNA or that aptamer that specifically binds to that target thing that we are interested in, right? For example, the hemoglobin, cholesterol. Men are interested about knowing about their PSA, prostate-specific antigen. There's there's a whole list of things that are known to have relation with the disease. We want to look for those molecules. So we need to have a recognition element, a molecule, organism, bunch of cells, DNA, proteins that bind to only that thing that we want to recognize. So that's the recognition part. Now, once you recognize the binding occurs, or whatever way we do recognition, it may be that it binds and it releases something, maybe. And maybe it's the binding that results into oxidation. Maybe it's the binding that results into reduction. Whatever the mean, that recognition, we should have a way of translating that event into something. We should have a way of transducing that into a measurable signal, right? Now that signal can be maybe current, like in the case of glucose sensor. That recognition may be emission of light, maybe we are using quantum dots, or maybe just simple fluorophores, simple fluorescence molecules. That recognition may be change in capacitance, that recognition may be any signal that can be measured, right? And then you have that front end thing where either you get a number or maybe you get a buzzer gong or whatever. So that's the outside interface with human. But these two things are the most important thing for a biosensor, right? It wouldn't work unless we have these two things. Why is number one important? Why is recognition important? What if it doesn't recognize? What happens? What do we call that situation when it doesn't recognize, or it recognizes something wrong, which is it is not supposed to recognize?
So then
Yeah, go ahead.
False positive, sir?
False positive or false negative, right? False positive, yeah, if somebody gets false positive, he or she gets tested, gets more results, and then ultimately we find out that the thing was not there, right? End goal is happy. False negative is much worse. False negative means that's human nature. A person will say, oh, I don't have it, then go home and continue with the life. But the thing was there. Whatever is ultimately going to become a bigger problem, right? So that's the recognition part. And transduction is the signal or the change in that the binding. We need to have a way that we can get rid of the noise. We can get rid of the presence of other molecules. It should be able to measure and identify that small recognition that may happen with very small delta, very small change in the number of or the concentration of the molecules of our interest, right? So one is related to selectivity. The second one is related to sensitivity, right? Sharam, kind of sleepy today? It's okay.
So all good, but just not feeling well.
Okay, okay. Yeah, that's why I felt. So okay, we are about to end. So what are we interested in detecting? It can be molecules, proteins, DNA, RNA, lipids, amino acids, a whole bunch of things. It may be complexes, right? Which would be pathogens, red blood cells, white blood cells, bacteria, organelles. It may be toxins, hormones, drugs, metabolites, whole range of things that are that is of our interest. For all of those things, we have to know exact recognition element, right? So, and we need to know what is the normal concentration or normal range, and what would be the range that is found in people who have the disease, right? So that that blood report you get, and they have the range, and then if some number is out of the range, this is nothing but the recognition and transduction giving us higher numbers, right? And from the data of human trial or data from normal people, we know what should be the range, right? Why do we want to detect it? It goes back to those 3 slides before: food, biotourism, biomedical, clinical, environment, public health and safety. So there's always something new, which with our knowledge we are finding out is maybe harmful, maybe implicated in some disease, maybe it's known that the exposure to higher dosage can be toxic or harmful. So we are finding new information. So we are becoming more we want to have sensors that can do that for us. Now, before we stop, here's a quiz. Name the biosensor that does all these things. It can avoid bad food by its odor. It can recognize people just from their maybe signature.
The nose.
Nose. Ahmed Imran brought the nose in. I didn't mean like you brought your nose in. I'm just saying, yeah, that's right. So think about it, that's a biosensor, right? So it helps us do many of those. So good job, Imran. Now, if I were to break it down as an engineer, that's how we do. So there are target molecules, there are recognition elements. There's transduction, there's a detector. And display is what our brain tells us to do, right? And then end user is we would move away from that toxic or place where there's bad smell. So if I were to break it down into block diagram, I can exactly put the features of a nose into that, right? And looking at nose closely, analyze the molecules in air, perfume molecules, bad molecules, volatile molecules, bad smell molecules. Recognition, we have receptors in the nose where they bind at the end of neurons. And those receptors, they convert that binding into an electrical signal that fires up those neurons. That firing travels, gets to the brain, and there is message that something needs to be done, right? So just and we'll get more into this, that biosensors, how do we design, what may be different functions of a biosensor, because that's all you see in many of the KPRs where you see application of those things. All right,